ecology, wrapping up the topic that we began on Tuesday of this week. I want to talk about ecology, that is to say, distribution and abundance of organisms. In particular, what is it that causes one lake to have a higher abundance of a particular fish compared to another lake? Or what is it that causes the same lake to have more fish at one time versus another? So that's a sort of what I call the fundamental question of ecology. Why is there more of this species in one place versus another? Why is there more of this species at one time versus a different time? Why? What is it that causes a difference, a specific difference in abundance? So we're going to kind of start with that question. I just have one small thing to do over here. All right. Okay, so to introduce this topic, I would like to share my screen. And if everything went smoothly, I think it's this PowerPoint. Ah, beautiful Mille Lacs. You know that giant lake right in the center of the state of Minnesota here? And you'll recall we are down here in the Twin Cities, Ramsey County, Hennepin County, Minneapolis, St. Paul, right there. So here's Mille Lacs. And Mille Lacs is a great big wind-swept lake. For a lake this size, it's huge, right? For a lake this size, it's not that deep, actually. And it is a prolific, what's the right word? A benign, a positive environment for walleye. Walleye, this fish here, a big member of the perch family, breeds naturally and prolifically in Mille Lacs. It's a natural walleye fishery, as they say. The walleye are there not because they're stocked by the DNR, but because they naturally reproduce there. And it's really because, in part, it's a relatively shallow lake with lots of islands underwater that are not too far from the surface. And when it gets really windy in the summertime, the lake mixes all the way down to there. And that's where the walleye breed. Like many members of its, the perch family, walleye lay their eggs in the springtime on in the shallows, on gravel, sand, or even attached to plants, draped upon the plants. And one of the biggest problems that fish have is having their eggs survive in the springtime. And one of the biggest challenges that fish eggs face is suffocation. You know, if they get buried in the mud, and there's any kind of natural decomposition going on in the mud, then the mud will become anoxic. And if the eggs are buried in even a little bit of mud, they can run out of oxygen and die, because, you know, there's a little fish in there that needs oxygen to live. So because Mille Lacs has lots of these sort of shallow places out in the middle of the lake, and they're not super shallow, but compared to the diameter of the lake, they're pretty shallow, and the wind will mix all the way down, bringing oxygen to them all winter long. So they're able to reproduce well in this lake. And that's true of many of the other famously good walleye lakes in Minnesota are, like here's Upper and Lower Red Lake up here, great big lakes that have large areas that are not too deep. So the walleye population, and we will talk about those things I just said again later today. And I will mention that the origin of Mille Lacs, part of the reason that the lake is so big is because there's a river that flows through it that's partially dammed at the outlet by glacial deposits. Yeah, so it's kind of cool in origin. Well, we looked at the walleye last week on Tuesday. And of course, the walleye population is very important in Minnesota, so there are good records of its population, good assessment. And this is kind of some DNR estimates of the total walleye population. And you can see that it dropped precipitously. This is just in the 20th century. These records go back 21st century. These records go back farther. But you can see that it dropped dramatically, seemed to rebound a bit, and then drop further. So you can see that this would really be cause for concern. So when we talk about fish ecology, I'm going to go write some stuff. I'm going to do something experimental here. Since we're just starting class, we'll see if this works. I'm going to now switch to a whiteboard. Can I do that? I think I have to stop sharing and then share something new. Share a whiteboard. Let me just give this a try here. Do we see the whiteboard? No. Here we go. So yeah, we're gonna try this. This is like, I'm gonna treat it like a literal whiteboard. So let's say that we're faced with what I would call the classic question, the central question in ecology, or the most fundamental Okay, that didn't work totally well. Question. I don't know if my handwriting's any better this way. Yeah, in ecology. Don't worry, my handwriting will get better over time, but you gotta practice. Okay, what's the most fundamental question in ecology? I would say it is this: why are there more walleye in Mille Lacs than in, I don't know, some other lake that you could name like Phelan? Or similarly, why were there more? I guess I write better on the whiteboard, but I'm gonna try. Here it is. In, let's see, what year did we have? 2018. Dan, in I forget what was that year, 2016? Was that 2016 was a low year? Right. I know that handwriting is not very good. I'm going to work on that handwriting. So what do I mean by this being the fundamental question of ecology? Well, what I mean is we got a question of, abundance. Why is there a change in the number of walleye? Comparing two places. There's one place versus another place. Right, so why are there more walleye? And we have our species here. Ooh. Or walleye in Mille Lacs in one year versus another. Or, you know, the parallel question would be this one: why are there more walleye? And the thing that makes this different, this a different question, the same question, but a different question, would be because we might compare 2018 to 2016. In other words, can you see this as I type? Why are there more, you know, some particular species? What happened there? At one place, type place or time than in other place or time. And I think I can make this bigger, although I don't see, you know, command format. 24, there we go. Right, so this I would call the fundamental or the central question of ecology. Why are there more of something? You know, typically we think of one species at a time. At one time, compared to another time, or at one place compared to another place. And I think that for me, this that we're looking at is, in this particular case, is walleye. And I'm gonna shift to the real whiteboard because like that was fun, but you can express your opinion on that at any point if you want. But I'm gonna just shift to the whiteboard here. And I would say that the answer to this central question of ecology is gotta be sort of at two levels. First, primary, and then sort of more secondary. So why do some places have more whatever than another? Why does Mille Lacs have more walleye compared to Lake Phalen? Why does, you know, anything you want? Why are there more pine trees in northern Minnesota than there are in southern Minnesota? You know, why are there more bald eagles in Minnesota in the summer compared to the winter? You know, could be any species, could be any two places, could be any two times. But no matter how you look at it, the primary answer to this is for primary population parameters. And people would say, well, the reason that there's more in one place versus another, one time versus another, must be a difference in one or more of the four primary population parameters. And what those are is those things all add up to combine to make little r. And those things are the birth rate minus the death rate. And those two things, if you treat them as, you know, exponents in a continuous growth and exponential growth model, those two things add up to form little r. Little r you know, where little r is defined as the natural logarithm of the net multiplication rate, right? That little r, the little r of e to the r. Well, the birth rate, sort of the instantaneous per capita birth rate, the instantaneous per capita death rate, people usually stop there, often stop there, but it also includes the immigration rate, and the emigration rate. So birth and immigration add individuals to a population. Death and emigration lose individuals from a population. If you want to think of it in a box and arrow kind of way, in a flow diagram kind of way, you're going to have the number of individuals here, and the two things that add individuals are birth and immigration. And the two things that remove individuals from this population, the only two ways that individuals leave a population is either by dying or by leaving, that is to say, emigrating. Emigrating. We have parallel structure here. Births, immigrants, deaths, and emigrants. All right. So in the most fundamental sense, the number of individuals in any population, in a population at any time, is equal to the number of individuals one time step earlier, and you choose a time step that's appropriate for your population. I mean, you know, if it was fish, you might be using years. If you're looking up birds from season to season, you might use months. If you're looking at populations of bacteria, you might use hours or even minutes. You know. But one time unit has passed. And the rest of this equation is plus something minus something plus something minus something, because there's only four things. And so it's like a really simple equation, right? So the number of walleye this year is equal to the number of walleye last year plus however many walleye are born minus however many walleye die plus however many walleye immigrate into the lake minus however many emigrate out of the lake. And, you know, it shouldn't be surprising, but this is where these letters come from, right? Births is, B is from birth, D is from death, I is from immigrants, E is from emigrants. Okay, so that's like some very, very basic ecology. I think I've mentioned, I think this is, you know, the central question of ecology, which I didn't write up there because I wrote it on the little scribbly whiteboard. And this is R here, right? And this is R of, you know, n sub t equals whatever, n sub 0, e to the RT. No, it's that little r. All right, so this is just some basic ecology. We're talking about the number of individuals in a population changing over time. And you know that a population is a group of individuals of the same species, right? Any questions about this? Because I'm gonna, well, let me tell you what your discussion question is, and then you can ask me if you have any, then you can ask me if you have questions. All right, so the question that I have for you is this. Am I sharing this? Here we go. Okay. So, of course, we're very concerned by the decrease in the abundance of walleye. And we're really happy that the walleye have rebounded. Well, maybe not you personally, but the state of Minnesota is very happy. So the question I have for you is about this rebound here. I would like you to think about the walleye and why there could be so many more walleye in 2018 compared to 2014. I mean, look at that. It's like more than twice as many. So my question for you is to come up with as many different reasons as you can think of to explain why the walleye were more abundant in 2018 than they were in 2014. So what I'm going to ask you to do is I'm going to put you in some breakout rooms, and I want you in the breakout room to just brainstorm. Appoint a chair, appoint a note taker, and think of as many different reasons, as many different things that could cause this difference in abundance. What could cause this difference have caused this difference in abundance of walleye? And then we're going to get back together and we're going to do something with your answers. So I will ask groups to give me their answers out loud. Okay, so I'm going to put you in a discussion group. In your discussion group, I want you to come up with as many different, appoint a chair, appoint a note taker, and come up with as many reasons as you can for what might have caused this increase in abundance of the walleye in Mille Lacs. Now that's your assignment. Do you have any questions about this assignment? All right, I've got, what do you think? Which is better, 3 people in a room or 4 people in a room? Anybody got an opinion? Oh, some people say 4, okay. So I will recreate these rooms. So let's have rooms with 4 or 5. Zoom is gonna make rooms with either 4 or 5 people in them. And Jeffrey's useless, so I'm gonna move somebody into that room. Sorry. Okay, so now we have 4 rooms with basically 4 people in each of them. So get together in your room and come up with as many possible explanations as you can, okay? And I'll give you, I don't know, 5 minutes or 10 to do this, something like that.
Oops, hello.
Hello.
Actually, I could be the note taker.
Okay. I feel like there's a lot of factors that would go into the walleye population.
All right, just name them all.
Maybe there's fertilizer runoff or salt runoff that goes into the lake and affects like the algae. Because if you've got too much fertilizer, then you get a lot of algae that goes into the lake, which can then suffocate the fish. Um,
Does that look right?
yeah.
Okay. Um, could people overfishing be a cause too?
Absolutely. Yeah.
Yeah, overfishing is definitely a factor. I know they set limits a couple years ago, so I think that might actually be what the increase was in 2014. So definitely overfishing.
Were you gonna say like not enough nutrients?
Yeah. You
Yeah.
think? Okay. I was just trying to like think of a reason as to why there wouldn't be enough nutrients, and it kind of just like reminded me of what you said earlier.
Yeah. I mean, it might also be due to like an invasive species going in. That happens a
Yeah.
lot.
I don't know if I'm still on this right.
It could be also like water. I'm trying to figure out how to phrase this one, but you know, if a lake loses water or if it has like some sort of change to it, then it might affect the fish within the lake. Because sometimes you need like a certain temperature or a certain amount of light in order to
Yeah. I
thrive and
know
water quality.
Water. Okay.
Yeah, like water clarity.
Clarity. Yeah.
Water clarity.
Yeah.
Clarity. Well, all those factors, yeah, temperature and clarity. Yeah.
Um, could we assume that 1 year mating was bad, and so there was less births and more deaths?
Yeah.
Totally.
Do you guys know if Mille Lacs is connected to other rivers or stuff?
I can look it up, but I'm not sure off the top of my head.
I
I
was
think
just gonna.
I have a cabin on the Lax actually, coincidentally, but I don't. Yeah, I think it's connected to like a really small. I think it's connected to the Rum River, but I don't think that there's many like major waterways that enter or leave it. I think there's just a small river, creek, or some stream. Yeah.
Okay. Yeah, I was gonna say something like immigration, but in that case, then probably
It
not.
could very well be like someone said, an invasive species, and I know that like boats can get
Zebra
like
mussels.
zebra mussels. So I'm not sure if
Yeah.
there's any like invasive species like that from boats in lax, but that could very likely be something. So yeah, invasive species or yeah.
You can also just have like flea problems.
What's
There's
that?
invasive fleas that will go into lakes, and that'll affect it.
Mm.
Invasive what? Sorry.
Fleas,
Please.
like bugs.
Oh. I'm just going to put invasive species again down here.
I suppose like predation, like other predators that actually may I don't know what eats walleye. I think muskie actually makes I don't know. Or of course their food sources too, like what they eat, food chain. It's all kind of obvious though, but
Okay, I'm going to go ahead and save us. If she asks us to speak on it, are you guys cool with talking about it? Because I'm not quite familiar with all this.
Yeah, for sure.
All right, looks like we've got everybody back, or almost everybody. So let's see if we can get some answers. Now, what I'm gonna do is I'm gonna write some answers on the board, and then I'm hoping you will see that they kind of fall, your answers kind of fall into different groups. Now, there's 5 categories of ecological factors that can cause a difference in abundance. And I'm hoping that all 5 of these will emerge. But anyways, that's where we're going to go, and hopefully you'll see this. All right, so let's get started. How about room 1? That's Brett, Kat, Tiffany, and Jude. What was your best one?
I guess we didn't declare a best one, but could it be from people overfishing?
Okay, so why were there more in 2018 versus 2016?
I guess we would assume more people fished in 2014 than 2018.
Okay, so there was a decrease in fishing.
Yeah.
Yeah? Okay. So I think I'll put that over here. Okay, so there might be more in 2018 than in 2016 because there was less fishing in between. The amount of fishing people did decreased. All right, good explanation. How about room 2? That'd be Brianna, Desiree, Liz, and Dolly, James. Well, it's a powerful room. Well,
So out of the reason we came up with, I guess one we'll share is that potentially there was less predation in Mille Lacs, so the age 0 to juvenile fish had more time to mature to adulthood, and then the population was increasing.
who do you think was doing the predation?
Muskies and like northern.
Okay, so less predation by, well, let's say pike. So northerns and muskies, not people. So the first one's about people, and the second one is about piscivorous fish. All right, good answer. Okay, let's go to room 3. Abdul, Keenan, Sam, and who else is in this room? And Zaid.
Okay, so we thought that, after like the population died off in like 2014 or the massive decrease, there was the lake was stocked in 2018.
Ah.
Yeah.
All right. I'm gonna put stocking over here. And by stocking, we mean humans putting fish into the lake. So human assisted immigration. That's what we mean by stocking. And there was an increase in the stocking rate, maybe from 0, yeah, but in any case, an increase. All right, good answer. Okay, let's go to room 4. That'd be Hannah and it's not scrolling. Hannah, Jeng Lee, Sam Thomas, or Saul? You have an answer that's an idea that's not up here yet.
Yeah, so another one we came up with was a possible decrease in emigration by the fish. Maybe the outflow was blocked in some way, preventing the fish from leaving the lake.
Sure. Yeah, maybe the outflow was blocked and this caused less fish, fewer fish per month or per week or per day or whatever, fewer fish to leave the lake. All right, we got 4 ideas up here. These fall into 2 categories. So I'm looking for ideas that are not on the board already. Let's go back to room 1. Brett, Kat, Tiffany, and Ju, you guys got something that's not up here?
We brought up invasive species. I don't know if that falls in with predation.
But so like what kind of invasive were you thinking?
Like zebra mussels,
Okay.
or water fleas.
Yeah, zebra mussels have invaded Mille Lacs. Why would the zebra mussels, this is a question for anybody, why would zebra mussels influence the walleye population?
From what I have known about zebra mussels is they like to eat up different nutrients. And so there might be something, and I'm not sure if like walleye use those nutrients or if they're eating plankton within the lake that the plankton use to eat those nutrients. So if you have a decrease in the plankton because of the zebra mussels, then you're also gonna have a decrease in walleye.
So zebra mussels are filter feeders. They eat things that are in the water. We use the word nutrients to mean things like carbon, nitrogen, phosphorus. But they certainly eat plankton, which contain nutrients. And because there's less plankton, less zooplankton, if there's less zooplankton, there's going to be less planktivorous fish. And the planktivores, the little fish that eat the zooplankton, those are the food that the walleye eat. When walleye are little fingerlings, when they're tiny fish, they eat plankton. But walleye grow pretty rapidly. No, that's not exactly true, but it's kind of true. A big influence of the zebra mussels is that they compete for the walleye. When the walleye are tiny, they compete with the walleye for food. And then when the walleye are bigger, they essentially starve out the food supply by eating the zooplankton that are eaten by the planktivores that are the fish that the walleye eat. Now, it's also true that by filtering the water, they actually increase the water clarity. And there can be some influence on walleye as well, because walleye do really well, actually, when the water is a little bit murky, because they got those great big eyes, right? They do pretty well when water is murky. So there's some evidence that by making the water more clear, that they're favoring the competitors for the walleye. in any case, there's definitely one way and possibly two ways that the zebra mussels reduce the walleye populations. Yeah, there we go. Okay, so that was a good one and complex too, because we're opening some doors to limnology here. Let's go to room 2, which is Bree, Desiree, Liz, and Dolly, and James. Do you guys got another one that's not up here?
I guess kind of tying in just to what you were talking about, we kind of brainstormed the idea of there was more resources as in like food sources for the fish. So kind of tying into the zebra mussel, there'd be a higher population of zooplankton for the fish to eat. And with more resources, usually species thrive better. So like food resources, I guess nutrients in that sense. Chemical nutrients, just more resources for advancement of the population.
Yeah, so actually you're talking about eutrophication.
Yeah.
And if you increase the nutrients at the bottom of the food web, it should increase it all the way up. And I just realized I went through this whole zebra mussel explanation, but actually the zebra mussels are probably bad for the walleye. But our question is, why are the walleye doing better? So I think we would have to say a decrease in zebra mussels. So maybe between 2016 and 2018, there was a decrease in the zebra mussels, and that's going to mean more zooplankton, which is going to mean more food for the walleye. Sorry, I had that whole thing backwards, just in terms of causing an increase versus causing a decrease. Okay, yeah, maybe the lake is more eutrophic. Maybe there's more nutrients washing into the lake, nitrogen or phosphorus from fertilizers or septic tank leakage, leading to there be more algae. More algae might mean more zooplankton. More zooplankton might mean more planktivorous fish. And those are the food for the walleye. Okay, these are all good ideas. These are all good ideas, but they're still only in 2 categories. So we're looking for ideas that might be completely different from these. Let's see, how about room 3? Abdul, Keenan, Sam, and Zaid. Did you guys have something that's not up here already?
Well, I just thought of this one. I don't think we wrote this down, but they just might have not dispersed to that area by that time. Like between like 2014 and 2018, there was like a massive
Inflow.
yeah, like this hadn't reached that area yet. Yeah.
Yeah, so maybe more walleye. Maybe it's a myth that they're reproducing really well in Mille Lacs. Maybe they're actually reproducing a lot better in, I don't know, the Mississippi River. And then what has happened is they've been swimming upstream and a great big school of walleye arrived in Mille Lacs in like 2017. And so there's more there. So there was some kind of like increase in the immigration rate. And that's a good idea. And it fits in this category. But you guys are doing a great job coming up with examples in 2 of the 5 categories. How about room 4? Hannah, Jang Lee, Sam Thomas, or Saul? Guys got an idea that's not on here? I guess not. All right. All right, I'll just open it up to anybody. Does anybody have an idea that's not on here? An idea that could explain why there were more walleye in 2018 in Mille Lacs than there were in 2016 in Mille Lacs?
The climate.
The climate.
Yeah.
What about the climate?
The temperature. Like maybe the.
You happen to know if the temperature's gone up or down?
Not off the top of my head, no.
Yeah, in fact, 2016 was globally the warmest year on record on Earth. 2020 tied 2016, or that's the latest that I've heard, which must mean that at least globally, 2018 was cooler than 2016. I don't know about the Mille Lacs area in particular, but it's because 2016 was such a record warm weather year. Let's say our hypothesis might be that 2018 was cooler and 2017 cooler. And when it gets too warm, the water doesn't hold as much oxygen and the fish don't do as well. They have a higher death rate, a lower reproductive rate. They reproduce in the springtime. So I think this would be more likely to affect things in the middle of the summer. So let's just say cooler weather, lower death rate. That's in a different category than these, and it's a different category from those. All right, so we've got, anybody got any other ideas that are not up here? Something completely different?
Can mating play a role?
Mating could play a role. Do you have an idea?
So because there was that drastic decrease, maybe they shared a lot of the genetic makeup, and so they didn't have viable offsprings.
Yeah. All right. So here's category 1. This is going to be category 2, 3, 4, 5. So there are 5 categories of ecological factors that can cause a difference in abundance. So there are 5 categories of ecological factors that can cause a difference in abundance. Or if you're comparing time, a change over time in abundance. A difference in abundance between 2 places, or a change in abundance over time. Different ecologists might group these differently. I think that a lot of people would say there's 4, that the 2 of these things belong together. There's other people who just say everything on the left is one category and everything on the right is a different category. I like 5 categories, and that's partly because there's other things like the 4 primary population parameters for which there are 4 things. So I find it a little easier to remember that in this, when I'm talking about this question, there's 5. I don't know. It's maybe like, okay, more about remembering it. So there are 5 categories of ecological factors that can cause a difference in abundance or a change in abundance. And the first one here is a change in dispersal. There's more in the population because the rate at which they're entering has increased, or the rate at which they're leaving has decreased, right? More are increasing because of humans putting them in. Fewer are leaving because the exit was blocked, or more are coming in for God knows what reason. They tore the dam down or something. So that's a big category that could explain a difference in abundance. And of course, this explains why there are more bald eagles in Minnesota in July than there are in January. And that's because the bald eagles go away mostly in the wintertime, and then they come back in the spring. Now, not 100%, but some bald eagles do stay. I mean, if there's open water, they'll stay and fish, right? But the abundance of bald eagles changes from summer to winter, really because of a difference in dispersal. Increased number coming in, increased number going out, and so on. Now, the second major category, and a lot of people combine these into one, but I think they're really fundamentally different, is that the organisms themselves may have changed in some way, something internal to the walleye population. And I would say evolution here, just to say, you know, a change in the frequency of alleles and or genotypes in a population. That's what evolution is, right? It's a change in the frequency of different types of alleles. And, you know, in other words, this is genetic change. And I would say, why does the population increase? Well, maybe the walleye have adapted in some way. Maybe the walleye have adapted. Maybe the walleye have evolved by natural selection in a way that has improved their fitness in mille lacs for whatever reason. Now, it could be that that there's genetic diversity and some of the genotypes are better fit and those ones were just better able to reproduce and, you know, and so on. Or it could be that people had been stocking the lake with walleye, but the walleye that were being stocked were not from Mille Lacs. They were from somewhere else in Minnesota, and they weren't really that well adapted to Mille Lacs in particular. And maybe the absence of that gene flow into the population, that flow into the population of less adapted genotypes, maybe that stopped. Maybe that stopped in 2014 or 2015, and those sort of less fit individuals have been replaced by better fit individuals, and the result is the population as a whole has done better. So some kind of evolutionary change in the population. The individuals that are alive now are on average more fit than the ones that were alive earlier, on average. So we're not talking about the individual changing, we're just talking about the whole population changing. So no individual walleye changes here. It's just the population as a whole has better individuals in it. And that's to distinguish it from this second one here, which is what I would call intrinsic factors. And this would be the walleye individuals are different in some way than they used to be. That the walleye individuals in 2018 were smaller. Maybe the walleye individuals in 2018 were smaller because they were younger, but if they're smaller, they eat less. So the zooplankton can support more of them. Same amount of productivity in the lake, same amount of zooplankton, same amount of feeder fish. But if the walleyes are smaller, the same amount of food supply can support more of them. So in other words, you know, the lake didn't change at all. It could just be that the fish, the walleye themselves are different. The walleye might be different genetically, a little bit more adapted or something, or it could be not a genetic change. It could be something like a change in the size distribution of the population, more small ones versus more big ones. Or it could be a change in like the sex ratio, more females and less males, because females are really the limiting factor for reproduction because they produce the eggs. Or it could be a behavioral change of some kind. I don't think walleye are particularly intelligent, but maybe somewhere, you know, in the early 20, during that long decline, maybe the bigger walleye learned not to take the bait. And so they're surviving better. Not because they've evolved, but because they've learned. So these are some examples here of things that could cause the population to increase. Maybe the walleye literally, I don't mean metaphorically, I mean literally learned how to not get caught. And that would cause the population to increase. These are all things that are internal to the population. They don't have anything to do really with the outside world. It's about the walleye changing. Either the walleye changing genetically or the walleye changing just phenotypically. So some people group these together and call these intrinsic factors. I think they're really different. Different enough that I would put them in two separate categories. Okay, I hope this has given you some ideas. Number 4, can you tell me what unites all of these things and separates them from like this thing? What are these, what is this category here?
I
Is
say-
it biotic?
Yeah, it's biotic.
Biotic, yeah. This category is biotic interactions. Interactions with other populations. Humans fishing them out. A pike killing and eating them. A complicated thing involving zebra mussels and the food chain. But the food chain is biotic interactions because they don't eat non-living things. They eat fish. Or another slightly simpler but food chain thing. More at the base of the food chain, more nutrients working their way up the food chain, more food for the walleye. This is all about a difference or change in biotic interactions. And then this last category, Hannah?
It's
Or a dual?
it's abiotic.
Abiotic, yeah. So this is abiotic factors. Change in the physical or chemical environment. The water is warmer. The water is colder. The water is more saline. The water is more acidic, less acidic, more saline, less saline, right? So windier, less windy. So this is non-living abiotic factors. So one thing that I would hope, regardless of which ecology category course you take, I would really hope that you would learn how to recognize an ecological question when you see one. And when you encounter an ecological question and try to think of answers, what could cause it? What could solve it? That you will think broadly about all 5 categories. You know, pest control. You've got an increase in ants in your kitchen or mice or bats in the attic. And why are there so many? There didn't used to be so many. That's an ecological question. Why are there so many? Why are there more, whatever, ants, mice, spiders, you know, bats? Why are there more now than there used to be? Well, it could be they've come in from somewhere else or they've stopped leaving. It could be they've adapted. It could be like, you know, they're at a different stage of their life cycle. like they've just reproduced, and that's why there's so many. Could be a biotic interaction, you know, Fluffy died, and now you got mice. Well, you know, or it could be abiotic factors. You know, the house is warm, the outside is cold, winter's coming, they've moved in. You know, it could be, well, maybe that's dispersal. So, and then when you think of solutions, you know, try to think of solutions in all of the categories. The first step is recognizing an ecological question when you're faced with one. And the second is really learning what these 5 categories are. Most people, I think, have a favorite category. And when they're faced with a question, they go to that category and they never leave it. Geologists, for them, it's pretty much all about abiotic factors. Anything changes and they look to, you know, the physical environment. It's warmer, it's colder, you know, whatever. Biologists often go straight to biotic interactions. Oh, it's a change in predation. It's a change in their food supply. It's a competitor. So I think that's kind of interesting, actually. But regardless of your bias, like whichever one of these you favor, I would hope that you would know what all of them are and be able to come up with an idea in each of them. This is one of my favorite exam questions, is to give you some kind of problem and say, what could explain this difference in abundance? Name the 5 categories of ecological factors and give me a reasonable hypothesis in each category. All right, well, that ends this part of the lecture. And I would point out that we'll do this again, probably, maybe even every day, I don't know. At least every week, because it takes practice to really think this way. And I forgot what I was going to say next. So we're going to leave this part of the lecture behind and move on to our next topic. Any questions about this? People who had ecology in the fall have a head start on this topic. Because as I mentioned, you know, I think every ecology class should cover it. But don't worry, you'll catch up with them soon. All right, well, let's move on to our next topic. Closely related topic, fish and ecology. So let's go to, here we go. Look at that. All right, so now I'm going to give you a little bit of a lecture that's really review, because you've read about this in Professor Kalf's textbook. And I want to mention, if you don't have a copy of the textbook yet, I was looking at Amazon, and there's a bunch of copies out there that you can get, used copies for, I don't know, 20 bucks or something, really cheap. And if you can't afford 20 bucks, so you don't have a credit card, or, you know, whatever, you don't like buying things over the internet, you know, I actually have an extra copy or 2 that I could lend you for the semester, and I can have the university mail it to you. You don't even have to pick it up. So if that applies to you, you know, send me an email. I'll send an email out to the class list so you can respond to it too. But there's really no reason not to have a copy of Professor Kalf's book for this semester. Okay, we're going to talk about fish and fish ecology. Anybody happen to know what are these guys? Recognize them?
They just look like perch.
They do, don't they? They are, in fact, yellow perch. Perca flavescens, which means yellow perch. And There it is, written more clearly than I can pronounce it. Here's the yellow perch. Now, one thing I'd like to review is just scientific classification here. We're going to be talking about fish. Fish are in the animal kingdom. They're in the chordata phylum, you know, everybody with backbones and spinal cords. And they are in a class. Now, not all fish are in this class, but all of the fish that we will talk about are in this class. I think that's true. This taxonomic class, the ray-finned fish. And you can see, perhaps you can see embedded within this name, actino, which means ray, and this one, pteri or pterig, which is a root word meaning wing, like pterodactyl or the wing finger dinosaurs. And ptera in this context refers to the fin of the fish. So these are sort of literally the ray-finned creatures. And then the suffix at the end just indicates that it's a class. So the different kinds of endings that you see on these words tell you whether it is a phylum or a class or an order or what have you. So we're going to be talking about the ray-finned fish. These are fish with real fins. And within that, there's two groups. There are fish that their skeletal components are calcified and therefore hard, like bone. And these are called the bony fish. And ostia, of course, is a root word referring to bone, as in bone versus cartilage. So all of the fish that we're going to talk about, pretty sure all the fish we're going to talk about are in fact in this subclass of the ray-finned fish class. Now within the class, the yellow perch are in the perciform order, and the suffix formas tells you that it is an order. It is an order named for its leading genus, the first genus that was officially named, and that is the perca. And all of the perch are in the perca, and the family of the perch is the Percidae, and the order of the perches is the perciforms. And of course, the name of the yellow perch, its scientific name, has two parts to it, a first and a last name, so to speak. The first is perca, the genus, and the second is flavescens, which is an adjective describing the noun, which is the genus. So the genus is a noun, grammatically speaking, and this part of the name is an adjective. And the species name is a two-word name, a noun and an adjective. So kind of literally, the scientific name is yellow perch. And that's its full name, yellow perch. And if you need to just talk about this word alone, you could call, that is called the species epithet, which means adjective. When you write a species name, it is important that you follow the correct format. The first time that you write the species name, you're going to write the whole thing out, perca, flavescens, both the genus and the species epithet. After that, like in a paper, the second or subsequent times you name it, you just abbreviate the genus, as is shown here. When you write a species name, the genus must be capitalized, and the species epithet is not. And the whole thing is either in italics, as is shown here, or it could be underlined. The scientific name is not in English. Therefore, when you type it up, you must put it either in italics or underline it, because it's not English, it's a foreign language, and that's the rule. So if you do that, you will always look like you know what you're doing, which of course is pretty important. You want to not only know stuff, but you also want to look like you know stuff, right? And when you're in ecology and you're dealing with species, you need to remember to italicize or underline, and you need to know which of these two to capitalize, because otherwise you look like you don't know what you're doing. So do that correctly, and that will help you in life. Okay, that's the yellow perch and a little bit of a lecture about scientific classification. We will talk more about the diversity of fish and the diversity of other taxonomic groups and the things that determine that diversity a little bit later in the semester. I rearranged the topics a little bit this year. Some of the reading questions for the fish chapter refer specifically to the diversity of fish. And we're not really going to talk about that today, but we will talk about that in the future. So that's a heads up on that. All right, so let's talk about the life cycle of the yellow perch. Okay, the fish life cycle is pretty typical for fish of all kinds of different groups. They are eggs. And this is a fertilized egg with like the baby fish growing inside the egg. This is not an egg cell anymore. It is a multicellular thing, but we still call it the egg. And I guess that's not surprising. I mean, a chicken egg is way more than one cell, and we call it an egg. So maybe that's not confusing, but I would just like to point out that when we talk about the fish egg, often we are talking about a diploid, multicellular thing like you see right here. You can see a tiny little fish curled up inside this egg. Let's see. For the yellow perch in Minnesota, yellow perch spawn in Minnesota in the springtime when the water warms up. It's maybe not as warm as you or I would like, but they tend to spawn pretty early in the springtime, right around the time that the ice goes out of a lake. In fact, because they often will spawn in streams that flow into lakes, they may even be spawning before the ice goes out. They might swim up the creek or up the stream, up the river a little bit and spawn upstream where the water is open and running, while the lake itself might still be frozen. Or after the ice goes out and the water starts warming up. So almost all fish in Minnesota spawn in the springtime. There are one family of fish in particular that spawn in the fall. Just as a trivia question, anybody happen to know which major fish family in Minnesota spawns in the fall, unlike all the others? Anybody?
Is it salmon?
It is. See, I knew somebody would know this. That's right. Trout and things in the salmon family spawn in the fall, not in the spring. Okay, so for yellow perch and most species of fish in Minnesota, they spawn in the springtime. For yellow perch, as I think I mentioned earlier, they lay their eggs on sand and gravel, or even like attached to vegetation, draped upon the vegetation in the shallows. So somewhere up to, 3-4 meters water depth. And walleye are basically the same way. Now, fish lay an astounding number of eggs. Your typical large adult female yellow perch is going to lay tens of thousands of eggs. 40,000 is a typical number that people kind of toss around, for kind of the typical number of eggs, fish eggs, produced by a female perch in the spring. That's a lot, 40,000. But by the time we reach the end of the summer, I think you'll find that 40,000 is maybe not too many. And that's because fish at every stage of that first year experience a very high mortality rate. For fish eggs, it takes, 20 or 30 days, a couple of weeks before they hatch into larvae. So these fish eggs are gonna, hang out in the shallows for 10, 20, 30, sometimes even 40 days, depending on the water temperature. And that entire time, they die. The most common cause of death, or the worst cause of danger, I'm not sure how you put this, a very important cause of mortality for these fish eggs is suffocation. Getting covered with silt, getting buried even a little bit, because they need to have oxygen going in and out of this membrane continuously, because there's a little live fish in there. They're also subject to fungal infection, things like chytrids and Uomycota, water mold, fish ick, if you've ever heard of these things, the sort of white fuzzy stuff that sometimes kills your aquarium fish. And then, of course, they're yummy and edible, and fish eggs get eaten by fish, because after all, fish are carnivores. And they will get eaten by other fish. They'll get eaten by other perch. The 1-year-old perch from last year is a danger to the eggs and the larvae and the juvenile fish of this year. And then there are other things like crayfish that scoot along the mud, and they will eat fish eggs too. So I would say that, generally speaking, the perch spend 10 to 20 days in Minnesota anyways, or 20 to 30 days, 2 to 3 weeks. Is that 3 to 4 weeks? As eggs. And then the eggs hatch. And I think you can see that there's a little larvae in here with 2 big eyes and a bulbous thing here. And here's a baby fish, a larval fish, still connected to the egg. And the egg contains nutrients that it is living off of, because it's not big enough to catch its own food yet. Now, a newly hatched perch larvae might be 5 or 6 millimeters long, so like half a centimeter, if you can picture that. And they do not have a calcified skeleton. They do not have strong fins. They don't have fins that are rigid yet. And the result of this is they can't swim. I mean, they can't really navigate. They can't control their movement in the water. And what happens to them is they get swept out into the pelagic zone of the lake, and they will eat plankton. Any kind of little plankton that gets near the mouth, they'll suck it in and eat it. So they basically live like zooplankton. And then like big zooplankton eating small zooplankton. And they'll spend a month or 2 as larvae. Now, it's a tough life being a fish larvae. They're at constant danger of starvation, right? They're not terrifically motile. They can't really control their movement very well. So they're not going to be they're not very capable of chasing food down and eating it. They really depend on bumping into food, in a sense. Food bumping into it. I mean, imagine if you could only eat things that bumped into your mouth. I think you'd starve to death. Unless it was like a perpetual food fight at your house or something. But in any case, larvae, their mortality rate for a population is somewhere between 5 and 25% chance. 5 to 25% mortality. In other words, on any given day, a larva has a 19 out of 20 chance of living and a 1 in 20 chance of dying. That's what a 5% mortality rate means. If a population has a 5% mortality rate, it means 5% of the population dies every day. So the mortality rate for larvae is between 5 and 25% per day, and that's a lot. I think you can see that if you started with 40,000 and you exposed it to even a 5% mortality per day, the total number would diminish pretty rapidly. Okay, so after like 2 weeks out in the, after like 2 months out in the pelagic zone, eating, not dying, getting bigger, putting on a calcium, calcifying the bones, developing some real fins, then hopefully what happens is, oh, and then here's the larvae, sorry, another picture. That's how big they are. And and they get called fry, by the way. Then they get their fins working, they put scales on, they calcify the skeleton, and they start to look like a fish, like a real fish. At that point, they're called a juvenile. They are a juvenile yellow perch. And the common name for these is fingerling. Although they're not exactly the same size as a finger, these juvenile fish can be anywhere from a centimeter or 2 long, so like the length of that guy's fingernail, all the way up. By the end of the summer, yellow perch will be somewhere between 5 and 10 centimeters long. So this is a really big fingerling. This is like as big as they get, like the end of the summer, 10 centimeters long. All right, so as a juvenile, yellow perch live in the littoral zone. They tend to migrate in and out in between the littoral zone and the pelagic zone, kind of back and forth, or if there is no pelagic zone, they tend to migrate up and down. And this is thought to benefit them by getting them to where the food is when the predators are least abundant, and then getting them away from the predators when the predators are the most. The biggest risk for these fingerlings is, I'm sure you can imagine, predation. You get to be this big and you look like a minnow, right? You look like food to a bigger fish. And all of those bigger piscivorous fish prey upon juveniles. In fact, cannibalism is a big problem for members of the perch family. So here you've got a baby perch at the end of the summer, and one of its biggest enemies is the 1-year-old perch and the 2-year-old perch from previous years. The mortality rate for juveniles is much lower than for the eggs and for the larvae, something on the order of 5%, 5 to 10% per day, which is lower than the 5 to 25% that the larvae face. And typically by the end of the summer, they might be about this long, maybe 5 to 10 centimeters long. Now, over the course of the entire winter in Minnesota, you expect somewhere between 75 and 99% of the fingerlings to die. You really typically would expect to see only somewhere less than a quarter of them will survive the winter. And winter survival seems to depend a lot on how big they are when winter starts. And the key thing is the gape. If by the end of the summer, it's been a good summer, warm, long, and lots of food, by the end of the summer, the yellow perch are going to be at the big end of their range, and they're going to have a big mouth. So here's this is more than this is not a first year, a young of year. This is like a 1 or 2 year old perch. But here you can see its mouth is open, and the diameter of the mouth limits the size of thing it can eat. You can see those teeth. Look at those teeth. These guys are carnivores. They eat fish. If the yellow perch start winter with a big enough mouth that they can eat little fish, they are much more likely to survive the winter. If, on the other hand, they end the summer kind of on the small side, they are much less likely to survive the winter. And it is thought that, yellow perch are basically sometimes people call them warm water fish, but that's not really true. It's not so much about the water temperature. It's really more about the length of summer. You get much farther north, like even northern Minnesota, southern Ontario, and the summers are shorter. They're just not as long. There's not as many days, and the water's not as warm. So the perch don't grow that as fast for as long as they do in southern Minnesota. And you reach a certain point, and it varies from year to year, but you reach a certain point as you go north where basically the perch just don't get big enough to survive the winter. Maybe some years they do and some years they don't. But that kind of thing is really tough on the overall population. So that's really what is thought to create the northern limit for perch as a species. Is kind of, the length of the summer and the survival of the overall population year after year. When the perch get to be this size, 1 year old and older, they tend to live primarily in the littoral zone, swimming in and around the plants. And they live primarily by eating invertebrates that are attached to the plants and eating things on the plants. So things like insect larvae, like dragonfly larvae, mosquito larvae, midge larvae, and things like that, leeches and other stuff, and also sort of larger zooplankton. They're not really filter feeders. They're kind of going for the bigger things that they can like see and bite. When they get bigger, like, 15 centimeters, 10 to 15 centimeters, kind of the bigger range, then they become piscivorous. They start eating fish, and they will eat little fish. So springtime, if they were big to start the winter, a couple of months into the spring, and they're big enough to start eating the newborn, the young of year of their own species. When are they adult? Typically, we say that the fish are adult when they join the spawning population, when they start reproducing. And that's not before 2 years of age, but pretty much always before 5. So somewhere in that 2, 3, 4 years is when the perch are technically no longer juveniles, but in fact, adult. That's what I want to say about the perch, I believe. And the next series of slides I have for you are research done on perch and other similar species. I'm just going to show you one or two, and we'll take our halftime break. This diagram here, and all of the diagrams I'm going to show you now, the charts, these are all from CALF. So you can read more about them in this chapter. This is chapter 26, and this is figure 4. And what this graph shows you is the growth of, individual yellow perch in different populations. 1 year old, 2 year old, 3 year old, 4, and so on up to 8. And this is the size of the perch. So this is like the average size in the population in centimeters. So you can see that by the end of the first year, in, kind of your typical lakes here in the middle, the 1 year old yellow perch range in size from, that looks like about 7 to me, more than 5 centimeters to less than 15. that's basically how big you expect one-year-old perch to be. Two-year-old perch in any of these given lakes, you can see that the perch grow rapidly 1 year, 2 years. By about 3 years, their growth rate is dropping off, but they continue to get bigger and bigger year after year. These lakes here are in Canada. I think Hertel is far north, Ile de la Prie and Memphremagog. This is the one that's got the sea monster in it. These are in Quebec and not that far north. And then you've got some other locations here, some of which I don't really know, like Lake Chad. It's kind of amazing too. These are, oh, some of these are different tilapia, sorry. These, some of these lakes are Lake Tilapia. Anyway, so looking at tilapia fish, these three perch. Okay, so that just shows kind of growth rates in different lakes. I think you would say that the fish in this lake are stunted, which is to say they're really significantly smaller than they could be. Even the 6-year-old fish in this lake are significantly smaller than they would be in other similar lakes. So if you were a fisheries manager managing a sport fishery where people like big fish, you'd be worried about this lake. You'd be worried about this lake having too many small perch. It's not that they're young, it's that they're, for their age, they are small. They are what they call stunted. I think that, yes. When you look at a perch population, of course, they're not all identical. They're not all the same size. And I'm going to show you 2 graphs from Lake Windermere in England, which is a lake we will talk about more than once because it is a very well-studied lake in the Lake District of England. This is Perca fluviatilis, which is a European yellow perch, extremely similar to the North American yellow perch. So what you see in this graph is in the springtime, sort of the range of sizes of the perch that they see. So in the springtime, you can see a range from, 4 to 8 for this year's newly born perch. But you'll also see a smaller number of bigger perch that are 1-year-old perch. And so when you harvest the perch from a lake, you will see these size classes. And this size class, this group of size classes here represents this year's young of year fish. And this over here represents the 1-year-old fish. And if you know how rapidly the fish grow, or maybe you don't even need to know that, you can actually watch the size classes move through time. So here we have in the early spring, in May, or late spring, in May, what you see is extremely small ones from this year, bigger ones from last year, and then a third size group from the previous year, the year before that. Month after month after month, the smallest ones get bigger and bigger and bigger. And then the 1-year-olds get bigger and bigger and bigger. So you can follow these size classes through time, watching them grow. And if you collected a sample of fish, to some degree, you can tell how many year classes, how many cohorts of fish are present. And it's pretty typical for some years to produce a lot of perch and other years to produce very few perch. So people talk about there being a strong year class and a weak year class, a strong cohort, a weak cohort. That's what I want to say about the perch as individuals, and then their growth rates and sizes and how that forms a pattern in the population as a whole. It's a little bit past 2:30 now, so I'm going to take a 10-minute break, go get a cup of coffee, turn the heater on so it warms up down here a little bit, and I will see you all in 10 minutes. 10 minutes. And I'm going to say hi to Christina. Back. Could be, Well, if you're back and ready to go, give me some kind of reaction. Yeah, party. Oh, I love you guys. Yeah. All right. Let's get started. What we're going to do now is just kind of wrap up the fish ecology section and move on to the lab. I'm just going to go through some of the key graphs, charts, data from this chapter. And I will point out that the lab we're going to do today is posted on D2L. So if your mind wanders while I'm lecturing or whatever, go to D2L, download today's lab. Okay, share screen. And let's go back to, et voila. Okay, so that was about growth of individuals and their sizes. This raises this big question. What determines the abundance of a species in different lakes? Now, fisheries ecologists don't usually care about the number of individuals. If we're talking about sport fishing, they do. They care about the number of big individuals because that's what sport fishermen like. They want to catch big fish. But the fisheries science has been primarily focused historically on food production. So the thing that they focus on is mass. How many pounds? Hundreds, thousands of pounds of fish. And it doesn't really matter how big they are. They just need to be big enough that you catch them in the net. And if they're big enough that you're going to catch them in the net, then the fisheries science calls that stock. So the stock of fish is kind of the catchable mass, typically mass of fish, or weight, the catchable weight of fish. Sometimes it's about number. Sport fishing is one example. Lobster fishing is another because people often pay by per capita by the individual for lobster. But anyways, the primary focus of fisheries science has been on the mass or the weight of fish, that is to say the stock, and how much mass or weight of fish you can harvest each year. And that is what they call yield. Yield is the amount of fish produced per year. Okay, so switching over to a kind of a fisheries science perspective, we're going to look at some data on what is it that determines the stock and the yield of fish. So here we have a graph. It's got biomass on the vertical axis, kilograms per hectare. And on the horizontal axis, we have total phosphorus. Phosphorus is often the limiting nutrient in lakes. So if you measure the total amount of phosphorus, that's a really simple way to get the total amount of, say, living material in the lake. So total phosphorus is often used as an indicator of the trophic status of the lake. Low phosphorus being oligotrophic, high phosphorus being eutrophic, and in between being mesotrophic. Those terms are explained in the Water on the Web reading, so you could go back and look at that. Okay, so here we have total phosphorus, which is kind of an indicator of trophic status, low to high, vertical axis biomass of fish, and in terms of mass per unit area. And what you see in all of these cases, pretty good positive trend. More phosphorus at the very base of the food web, more fish. And even in individual lakes, you can see this correlation. Here's a particular lake in which the total phosphorus was decreased, and almost simultaneously, the fish biomass decreased. This is pretty solid evidence that the total phosphorus is what controls the fish biomass. In other words, kind of a bottom-up control, the bottom of the food web controlling the mass at the top of the food web. What about productivity of biomass? There's a difference between biomass that's just sitting there swimming around unchanging and the production of new biomass year after year after year. If the stock is really big, but the productivity is low, you might go, oh, look at all these fish. And then you harvest them all, and then they're gone. If you want to keep harvesting a fish population year after year, then what you really care about is fish production. In other words, fish yield. In other words, how much biomass, how much fish weight does the fish population produce every year? High productivity meaning lots of fish biomass produced every year. What is it that controls that? Well, there's a pretty strong correlation between primary production, meaning photosynthesis, and secondary production, fish. And in this graph, you'll notice that it's a logarithmic x-axis. So this is 10 times this, right? The primary production of this lake is 10 times higher than the primary production of this lake. And similarly, this is like 10 times that, and so on. So this is a logarithmic x-axis and also logarithmic y-axis. But what are the two factors involved? Primary production, meaning photosynthesis, producing grams of carbon per square meter per year, versus fish production, kilograms per hectare per year. So this also kind of indicates that the production of fish is strongly correlated to production at the base of the food web, bottom-up control. Now, I want you to be able to learn, how to read tables and how to read equations. Now that might sound pretty simple, but obviously if some of you have discovered, you thought you knew how to do that, and it's actually harder than you thought. So in this textbook and in this course, we will look at a lot of graphs and a lot of tables of data. Let's see, so what we've got here is showing you in different research studies, Randall et al., Yurkin, Bachman et al., Downing, I think that's probably Downing from Iowa, Liang, Schlesinger, and so on. So these are all different published studies looking at different groups of lakes. In each of these studies, you can see how many lakes there were. There were 11 lakes in this study, N being a typical way to represent number. In this case, that would be sample size, number of lakes. In this study of Florida lakes, 66 lakes. On the left-hand side, we have the equation that they discovered when they measured a whole bunch of lakes, 11 lakes, 22 lakes, 65 lakes, whatever. And they were trying to see what factor determines the biomass in terms of kilograms per hectare, what determines production, kilograms per hectare per year, or catch. Catch is kind of similar to production, but catch is what you actually caught, whereas production tends to be what's inferred, even if it's not caught, like out in the lake. And here's the equation. And then here's the R squared, like how good was the correlation? A high correlation like 96 is a really strong correlation. A low R squared like 0.24 sounds like a pretty weak correlation. And it's going to be a number between 0 and 1. So let's focus here on the equations for biomass. Coming up with an equation that's going to determine the biomass of fish in a lake. And let's focus on this graph. You saw this graph before. This graph is showing the same data. So, for example, here we have a study by Randall et al. They looked at 11 largely North American lakes. Let's see, if this is Florida and this is the world, I think this must be the world lakes. Oh, maybe we'll pick another one that we're just a little more sure really is the thing we're talking about. Let's go to Yurkenay, 22 reservoirs in the state of Virginia. That's this data right here. 22 of them, and there's the line that fits. All right, so what is the equation for this line? The equation for this line, on our x-axis, we have a logarithmic scale of biomass. So x is going to be the natural logarithm. Let's see if I can type this here. The base 10 logarithm. On the x-axis, we have the base 10 logarithm of biomass, right? On the y-axis, talking too fast. On the x-axis, we have total phosphorus. This is also a logarithmic axis. So the y-axis is log of total phosphorus. So the equation that we're going to come up with here is going to be the logarithm of biomass is equal to the initial biomass when total phosphorus is 0. Total phosphorus being 0 would be somewhere over here on your graph, like right, well, you can't actually get to 0 on a logarithmic graph. You know, it always starts, because that would be like infinite. It's undefined. So a logarithmic graph never goes all the way to 0. But you could have Excel fit a line to this data, and it might easily give you an initial value of something like 1.07. And then it's going to give you the slope of this line. You can measure this yourself as rise over run, right? And the slope of this line, if I'm reading the formula correctly, is going to be 1.14. It's a little steeper than a 1-to-1 line times the logarithm of total phosphorus. This is the formula for this line on the left here. I'm not sure where I should put this, over here. And you'll notice I really just copied, the formula. Okay, get out of there. I just copied this formula up here. All right, so that's the connection between the numbers on this table and the charts, or at least this chart. And what you can see is that for this data set, in particular, the correlation is really pretty good, 0.75. These black dots really fit pretty closely to the line. But you may notice that the Florida lakes line, okay, I gotta stop annotating here. The Florida lake line, which I think is this one here, the one that has the lowest slope. See, this is the shallowest slope. So of these three, it's the one with the lowest number for a slope. Wow, I really don't think that these data points fit that line very well. I mean, there's a lot of triangles, but a lot of them are really far away from the line. I really don't think this line explains the pattern in those triangles all that well. And in fact, the R squared is very low, 0.24. of these three, the Florida Lakes has the steep, did I say this wrong? Of these three, the Florida Lakes has the least steep slope. It's the least steep. The Virginia data set has the steepest slope, the biggest number for slope. I would say the Virginia Lake set has the best R squared. And I would say that the Florida Lake set has the worst. And that this other data set, if in fact, I'm not completely sure that the world, so-called world lakes is the same, I think maybe it is somewhere in between. Okay, so when you look at tables and you look at charts, these are the things that you want to be asking. What's the X axis? What's the X variable, the independent variable? What's the dependent variable? What's on the Y axis? Is it a linear axis or a logarithmic axis? How good is the correlation? Is it a big correlation or a small? How many lakes are involved? How big is the sample size? If there's a lot of lakes involved, I'm more likely to believe that it's a reliable answer than if it's a small data set. I'm like, well, you know, a lot of lakes are really different from each other. It depends on which lakes you picked, you know? So you want to look at the sample size and think about it. Look at the R squared, the correlation coefficient. Look at what's being predicted and what's going into that prediction. All of these are based solely on phosphorus. Okay. I encourage you to read this text that I have copied here. Clear my dogs. You know, equations describing biomass, production in fish, and so on. So this is the caption for that table. I encourage you to read that. And I'll just kind of conclude this section by saying, when people are trying to predict fish yield, one of the most famous things that people have used is called the morphoedaphic index, which is like a body mass index. It combines two or more factors. It combines total dissolved solids, which is similar to total phosphorus, total amount at the base of the food chain mostly. It combines that with the mean depth as a way of predicting fish yield. So if you're interested in fish, I would draw your attention to the morphoedaphic index, which is kind of famous or maybe just classic. And in any case, everybody should at least read it and see if you can understand what it means. Of course, you know, the bigger the fish are, the fewer of them can be supported with a given food supply. So when in lakes where the fish are smaller, for all of these different species, regardless of what species you're talking about, if the individuals are smaller, did I say that wrong? If the individuals are bigger, sorry, moving left to right, if the individuals are bigger, there will be fewer of them in the population because smaller fish eat less. The same food supply could support more fish if the fish are smaller. On the other hand, it doesn't really matter whether the fish are big fish or small fish. You see these lines are mostly kind of sort of horizontal. They're definitely not all going like that. Basically, that doesn't affect the total biomass of fish. So when fisheries ecologists focus on biomass, they may come up with very different answers than an ecologist who's looking at abundance. So another factor that determines abundance of fish would be the size of the fish, which is what I would classify as an intrinsic factor. The final slide I want to show you is just kind of introducing the idea of diversity. How many different species of fish will you see? And that's what this graph is showing. And we're going to come back to this graph in a couple of weeks when we start talking about biodiversity. we will probably start talking about biodiversity when we talk about plants. But when we talk about biodiversity, we're also going to look backward at the fish and the zooplankton and the algae, everything that we have talked about up to that point. So when we get to the concept of biodiversity, we will come back and look at fish. And here are the references for some of the pictures, especially the perch pictures earlier. And I want to conclude with this picture from the DNR and turn our attention now to today's lab activity. Okay, so we got 3 members of the Asacidae, the pike and muskie genus. Well, family. Asacidae is the genus. They're both in this, say that right. They're both in the same family, Asacidae, and they're both in the same genus, Esox, E-S-O-X. Let me type that up here. Annotate. So these are both in the genus Esox, family Asacidae. And I don't even know that I can italicize, but it's time for me to learn if I can do that. I can. Here we go. Oh, I can only italicize the whole thing. Oh, well. Okay, so muskie, northern pike, hybrid of the two, tiger muskie. Got that picture in your mind? And so here's my question for you. I'm gonna ask you, what is this fish? Clear my drawings, there we go. Okay, you ready? So I'm gonna show you a fish and I want you to tell me, is it a muskie? Is it a northern pike? Or is it a tiger muskie? Tiger muskie, that name goes with that fish. I tried to kind of scale them. Muskies are the biggest. Pike have a wide range of sizes, but basically they're never as big as a big muskie. And tiger muskies are a hybrid between the two. So I'm going to show you a picture and I want you to guess, what is it? Is it a muskie? Is it a northern? Or is it a tiger muskie? Okay, you ready? Got those 3 images in your mind? Ah, here it is. Okay, name that fish. Take a look at it, make a guess, put it in the chat. Is this a muskie? Is this a northern? Is this a tiger muskie? And the votes are rolling in. And oh, yeah, we're getting quite, nobody thinks it's a regular straight up muskie. It looks like the votes are kind of evenly divided. Okay, look at this, it's got a white underbelly, fine. It's kind of greenish, fine. It's got little white spots. You see how the white spots are kind of parallel to the body itself, kind of dotted along like that, a little bit weirdly underneath here, complexly. Right, so you see those light spots kind of parallel to the main body length. That's our key. And now that you've seen that, care to vote again, everybody? Feel free to change your opinion. Put it in the chat. What do you think it is now? And yes, I believe that, yeah, got some more thinking here. Yeah, some people are changing their votes. That, in fact, is a northern. You see those white spots kind of parallel. The muskie is more like dark spots on a light background. And the tiger muskie is called tiger because it really has these sort of perpendicular to the length kinds of stripes, that the spots are sort of oriented in these almost vertical bands. Yep. Great big monster northern pike. All right. There you go. That is your introduction to whatever that was, fish ecology. And now we're going to turn to the lab. I'm going to, don't save any of that stuff. And, oh, I guess I stopped screen sharing too. All right, here's the idea. There's a lot of evidence from fishery science that productivity at the base of the food web, the base of the food chain, is what determines abundance at the top. And I think that this is really very well established, that if you have nutrients and light, and by nutrients we mean phosphorus, nitrogen, and other things. This is going to control the amount of phytoplankton. This will control the amount of zooplankton. Right? This is going to control the amount of planktivorous fish. Now, the planktivorous fish, I got to change markers here. It's going to include, young of year perch, and also a lot of other fish that are always small, and sunfish and stuff like that. So this is going to control the abundance of planktivorous fish of various species. And then you're going to have your piscivorous fish. And I'm going to put walleye here as our classic piscivorous fish. It's the one that people are worried about. Because the thing that worries people is if you add muskies to a lake, what is it going to do to the walleye population? Muskies eat walleye. Muskie eat adult walleye. Muskie eat juvenile walleye, and they eat the little fish that walleye eat. So they are both a predator of walleye and a competitor of walleye, eating the same thing that the walleye eat. So I think that people have good reason to be afraid. If you took a perfectly ordinary good walleye lake and you added muskie to it, people are afraid, in some cases, that this will harm the walleye population. And there are people who live on lakes or fish at favorite lakes where the DNR has proposed to stock walleye, to stock muskie, who have vigorously opposed the stocking of muskie. And the DNR says, oh, no, the muskie don't really eat a lot of walleye. They mostly eat blah, blah, blah. Okay, so who's right? Science has the answer. People got one opinion, people got another opinion, they're arguing about it, both sides, it seems logical. Science steps in and tests their hypotheses. So in this lab today, what I'm inviting you to do, and you're going to have to download the lab to see the lab instructions. I hope that they're clear. I wrote them yesterday morning because we needed a new lab today because, we can't meet in person for the fish lab. So here's a new lab. And we're going to test this very important hypothesis. In order to do this, what I want you're going to collect your own data. Am I talking too fast? Are you guys with me? Uh-oh, I left everyone behind. Are you still there? Hello? Everybody there? No? Okay.
Yep.
And you have access to the lab instructions, right? So the lab instructions are on D2L. And you're going to need them. And I'm just going to show you the part that I think needs explaining. What you're going to do is go, oh, Jeffrey says I need to share my screen. Thanks, Jeff. Share my screen. Google Chrome, here we go. Bam. Okay. So what you need to do is go out to the Minnesota DNR Lake Finder. And that's Lake Finder. In fact, I don't even think you need to put in Minnesota and DNR because I think there is only one Lake Finder. But anyways, go to Lake Finder. Yeah, you might have to like click around a little bit through the DNR webpage. But here it is, Lake Finder. Jeffrey. There we go. The Wi-Fi is a little slow in the basement for some reason today. Okay, so here's the Minnesota DNR Lake Finder. You can use it in a variety of ways. I'm going to show you one way and invite you to explore other functions on your own. What you can do is you put in a lake like Lake Phalen, and you might put in the county. For every lake about whom you need data today, I have given you the name and the county. There happens to be only one Lake Phalen, I believe, in the state of Minnesota. So that's all I have to do. Here it is. You can see there's only one. Here's the name of the lake. Here's the lake ID number. And as I think I explained last week, Ramsey County is County 62. The first 2 digits are the county number, counties in alphabetical order. And then the next 4 digits are the lake number. This is lake number 13 in Ramsey County. Here's the name, Ramsey County. Sometimes there's more than one lake of the same name in the same county. Like there's like 2 or 3 Round Lakes in Ramsey County. And so they also put in the nearest town. When you're reporting on a lake in a lab report or any other thing like a lab report, you really want to include the name of the lake, the county of the lake, and if you're in a state like Minnesota where the lakes have numbers, the lake number. So you click on the name of the lake and you get all kinds of cool information about the lake, both in the DNR website and also in the Pollution Control PCA website. Here's a picture of the lake and you can, zoom out or zoom in. You can get it in satellite view or in topographic map view, kind of cartoon topographic map view. You get your basic information about the lake, area, shore length, littoral area, kind of a summary of what fish are found in the lake, and some other stuff that I always ignore. Okay, but I mean, you can.
Quick question, Virginia.
Yes.
Is that little lake that's right next to Phalen there, is that Round Lake?
That's Round Lake.
Technically, so is it considered part of Lake Phalen then?
No, it's a separate lake.
Okay.
Yep.
It just makes it look like it's one lake, like one big body.
Well, yeah,
is what it kind of looks like.
I mean, if you look at it from space, yeah. Oh, I'm sort of laughing.
But I mean, like when it has it highlighted, it just kind of looks like it's highlighting the whole thing as Phalen versus.
Yeah. Well, that's funny because somebody else said that in this class, but nobody's ever said that before in this class. So I don't know. I'm not really sure. I don't have a standard answer to that question.
But you know what I mean, right? Or do you see like what I mean there?
I guess so. I mean,
Like
maybe it's the way that the park. Yeah. Okay. Anyways, you raise a good point. All right. So there's Lake Phalen, and over here on the left, the information that you need is here in the Fisheries Lake Survey. In the Fisheries Lake Survey, this is now fisheries data. You can see a little description of the lake, blah, blah, blah. You can click on this menu and see all of the different years where the fish have been surveyed. Right, going back to 1979. So we picked the most recent one. And what we're interested in is, well, that's interesting. Well, what we're really interested in is neither of these, a beach seine or backpack electrofishing. What we're really interested in is just a standard gill net. So I'll go to the next year. So you want standard gill net. And we're really only interested in the walleye, which you can see right here, or if you wanted to, you could just narrow it down with the list. So what they do is they set out gill nets in a particular way of a particular size, and from this they calculate what is known as CPUE, catch per unit effort. And this is a way of standardizing gill nets. So like on one lake, they might have put out 1 net. On a different lake, they might have put about 10 nets. They might have used a bigger net or a smaller net, or left one up for 2 days and one up for 1 day. They could take their net data and then they standardize it so you can, it's apples to apples comparing lakes. And the standard unit is CPUE, catch per unit effort. That's a standard fisheries unit of measure, catch per unit effort. You can see that on Lake Phalen in this year that it was studied, they found 1.8 walleye in the gill nets, in the catch per unit effort gill net. The normal range for lakes of this type is like 1 to 6. The average weight of the fish that were caught was 1.5 pounds, and that's in the middle of the normal range. And I'm not sure whether count means the number of fish or the number of nets. I haven't really thought about that. Anyways, the number we're focusing on here is the catch per unit effort. So in the lab instructions, I give you a list of the 11 lakes in Minnesota that have been stocked with tiger muskies. There are 11 lakes in Minnesota that are stocked with tiger muskies. Now there are only 34 lakes in the entire state that have natural muskie populations. So the other 60 lakes have muskie because humans put them there and continue to put them there. So muskie are natural in only a very small number of lakes in Minnesota, 34. And hybrid tiger muskie are not really natural anywhere. I mean, they probably do occur because they are a hybrid between muskie and northern, but they're sterile. They can't reproduce. So, you know, you can't really, I'm not really sure you would ever say there was a population, natural population. Anyways, these 11 lakes they're all in the metro area, have been stocked with muskie, hybrid tiger muskie. And we know because tiger muskie are not naturally reproducing anyways, we know that the tiger muskies that are sampled in these lakes are the result of stocking. In the lab report, I gave you a list of 11 lakes, the 11 lakes that have been stocked with tiger muskie. And then I went through the 11 county metro area and I picked out 11 lakes that were kind of similar, but did not have tiger muskie or muskie. And the challenge is to determine whether the tiger muskie are having an impact on the walleye population. do lakes that have tiger muskie stocked in them have fewer walleye or not? Now, there's a lot of additional information that you could pull into your analysis if this was the kind of thing that interested you. This is a very simple question, and I've spelled it out pretty clearly. The data you need to collect can all be collected from the Lake Finder. Really, all you have to do is collect this data and then use a t-test. If you want to do more than that, I invite you to, but make sure you do at least that. All right, so I'm going to stop sharing. I'm going to draw a cartoon on the whiteboard. And the cartoon is going to be really simple. It's really just our hypothesis, right? Our hypothesis is lakes that are stocked versus not stocked. And here's our walleye in catch per unit effort. And you have to do that because, a bigger lake is going to have more walleye because it's bigger, but that's not meaningful. Catch per unit effort enables you to get something like that correlates to density, number per hectare. Our hypothesis is, what's our hypothesis? Which is going to have more? Like if the stocked lakes have an average here, what do we predict? This is walleye. What do we predict for the average number of walleye in not stocked lakes? Are we predicting it's going to be above, below, or the same? So what's our prediction for the not stocked lakes? Above, below, or the same? You could put that in the chat, make a guess. Based on this food web theory, pull up my chat here. So based on this fear, what do we predict? When we compare the walleye populations in stocked lakes to not stocked lakes, all other things being equal. Not stocked. Yep. Our hypothesis is that lakes that are stocked with the muskie are going to have a lower abundance of walleye than lakes that are not stocked. That the result of the stocking is going to be a decrease in the abundance of the walleye. Now, to support our hypothesis, we need to find not just that the abundance is lower, but that it's significantly lower. And in order to prove significance, we're going to compare the two confidence intervals. So 11 lakes is going to give you an average, a standard deviation, and a confidence interval. 11 lakes unstocked are going to give you an average and a standard deviation and a confidence interval. And then we use the rule of thumb t-test to compare these two. In this lab assignment, I ask you to make two kinds of graphs. This is a t-test type graph. The other type of graph is just called a dot chart. So this might be your average, and this is your other average, but every single data point is just there as a point. So you would have 11 data points here, and 11, whatever they are, 11 data points here. And this is walleye. A t-test chart really shows you, the statistical significance or not of the difference. But the dot chart actually shows you every single data point. And so you could see if maybe there's an outlier. You know, you could see if they're all basically close together except for one, and that drags the average up. It enables you to kind of see patterns in the data that might be hidden inside these calculated values. So you're going to make two kinds of charts, a t-test type and a dot chart. And I posted just a one-page instruction for how to make both of these in Excel. There's actually more than one way to make this type of chart in Excel, but I've posted a one-page sort of visual how to do it. Okay, that's the lab. My guess is it'll be pretty quick. I don't know. It's hard to say. So do you have any questions?
When are your office hours again?
My office hours are by appointment when you need me.
Okay, sounds good.
I'm in class all day, Tuesdays and Thursdays, so I'm not available on Tuesdays and Thursdays basically, but you know, otherwise I'm pretty flexible.
Okay.
So what I would suggest is that you go to the webpage and start pulling down some data. If you have a question, you can share your screen with me and show me what you're having trouble with, and that we just get started doing the lab right now. Any questions? Okay, well, just let's get started. And I'm just going to be here.
Hey, Professor Card,
Sure.
are we going to be doing the Q&A in like 10 minutes?
I guess we can do Q&A kind of anytime, but I guess I was thinking maybe just right at the end to make sure that if anybody's having trouble with the lab, we deal with that first.
Okay, all right.
Or if you have a specific question, you can email me.
Okay, that works too.
Yeah. And I'm going to check my email too right now if anybody has like questions they want to ask me, but not like, you know, see, I'm checking my email right now. So if you want to email me anything, you can do that.
Dr. Carl.
Sure.
Sorry, I didn't know what is Q&A is. It's a different quiz. Do we have or?
That's just if, I think some people had questions about some of the questions on the quiz that is now in the past. So quiz 2. And there will be a quiz 3 posted sometime tomorrow.
Okay.
So,
Okay.
yeah, that was just if people had questions about the previous quiz.
Okay.
Yeah.
Professor Card.
Yeah.
So you know how for Phelan, for the gear you chose gill nets, right?
Mhm.
So when I'm doing the first lake, Crystal Lake, the
Yep.
gear that it has.
Yes. You want gill net.
Okay, because I only see like trap nets or
Oh, well, all right. I didn't
Yeah.
actually check that these all had all of the data.
Yeah, the standard gill nets for Crystal Lake don't pop up until 2005.
Oh, okay.
So I don't know if when this data was collected matters for the lab or anything. If you want consistency on the survey years, or if you just want whenever the first standard gill nets show up.
Well, I think it would be important that the gillnet data be after the muskie were stocked. So does that show? That's a really good question. So this is Crystal Lake in is that the Crystal Lake in Hennepin, or is that the Crystal Lake in Dakota?
In Dakota.
Dakota. Crystal Lake in Dakota, Lakeville. Well, you'd think if they were stocking it with anything, they would be doing fisheries surveys, wouldn't you?
Yeah, so when I click on the lake fisheries surveys, the standard survey done in July of 2005 is the first one that offers standard gill nets, except they don't have walleye as a species option.
So they're doing this. Wow, this beach seine. Oh. So what's equivalent to gillnet? So by first, you mean the most recent one?
Yep, the most recent one would be the July of 2000.
And then they just got trap nets in 2011. Oh, wow, that's really interesting. I didn't expect that. Okay, so they're trap netting. When did they start stocking it with? I guess you could go out to the stocking survey to see. So maybe I'll just you can watch this or ignore me. So if you go to the fish stocking,
Looks like 2012.
so they started stocking them in 2012. So we really need fish data after that. Well, I wonder if that's going to be typical. I wonder if that's going to be a problem for any of the other lakes too. Targeted survey, standard survey. Standard trap nets. When did we say they started stocking?
2012.
2012, so it's got to be more recent than 2012 that we get something. Boy, I don't see any walleye at all on this lake.
Yeah, it looks like for Crystal. I've been clicking on some of the other ones. I haven't seen walleye pop up for any of them.
I saw it in 2000, and that's it.
I checked Orchard Lake, and in Orchard Lake they have walleye. I see walleye for Orchard Lake.
That's very interesting. I mean, maybe they, I wonder whether they picked these lakes in part because they don't have walleye.
And you wanted them in the gill nets, right?
Yeah.
Yeah. And if
So,
there
yeah.
are no gill nets, I think we're going to have to go for the electroshocking or the beach seining or something. Because it kind of looks like maybe they're doing away with the gill netting.
Yeah, for orchard, which is the second one on the list, they have it for electrofishing and gill nets. There's walleye when I check on Orchard Lake.
Yeah. Okay. Well, we'll have to see what we see here. Boy, that lake is only 9 feet deep. Or the
Crystal Lake is really small.
wow.
Or it's not deep. It's
It's
a
not deep.
pretty decent sized lake, but it's not deep.
Yeah. Yeah, I see. So
My aunt
yeah.
lives right off of the lake, so
Oh yeah? Cool.
yeah.
So it's almost 300 acres, but 200 of it is littoral zone. Yeah. The mean depth of 9, but a max depth of 37. Maybe they figured that it was deep enough. Yeah. Okay, so if there is no gill net data, then go for whatever they have.
So are we only looking at dates after 2012?
Well, that was for Crystal Lake, from my
Yeah.
understanding.
So you'd want to, if the most recent, these are all lakes that have been stocked since 2016.
Okay.
Or in 2016, they were reporting these as being stocked.
Okay.
But you can go to the lake finder and let's take another lake. What's another lake I should look at right now?
We could try Bryant Lake.
Wow.
I mean,
That's a really small, why was I thinking about Bryant Lake recently? Anyways, it's bigger than I thought. 200 lake. So we can look at the survey data. So it's 2017. And it's got gillnet data. And it's got tiger, muskie, and walleye. Go Bryant! Yeah. And it's got both. It's got just standard gillnet and. Okay, so Bryant Lake is good. I should probably make a note of this one.
Yeah, so just as we're working through this throughout the week, and I'm sure maybe you'll double check some things too. Are you not going to be too concerned then about like which surveys we use, depending on the availability of the information on the website, as long as it came in after after when tiger muskies were stocked in that particular lake? If my question makes any sense.
Yeah, your question makes sense. I mean, if this was a real study, then you would also want to include when it was stocked with the tiger muskies.
Yep.
And when your survey data came from.
Mm-hmm.
As I picture that in people's lab reports, that sounds like a whole nother data table.
Yeah.
Which would be good to include, but it's really more than the minimum.
Okay. Yeah, I was just curious if you wanted us to maybe just like include that as a note in the
James,
discussion.
So James, what I want you to do is include a table with which survey data you're using, because you'll get a bonus point for doing that, and you deserve one.
I appreciate it.
Yeah.
Quick question. So if there are other lakes like Crystal that pop up that just don't really have sufficient walleye data, what really should we do with them? And just not include them in the lab, or?
Well, they're going to be, if they don't have any walleye even before the stocking, then I think it makes sense to leave them out. So if they don't have any at all reported after the stocking, I think you would go back and look and see if there's ever been any reported. Because it doesn't really make sense to look at their effect on walleye if there really were never any walleye there.
Well, I guess more of what I'm asking is, like, we know Crystal Lake had walleye because back in like 2000 they reported that there were walleye, but I mean that was 20 plus years ago now. I don't know if it'd be super relevant to, especially the data we're collecting on that's kind of about after this 2016 stocking. So,
Oh, I mean, don't you think it's possible that the muskies eliminated the walleye?
I mean, yeah, I guess it's possible.
Yeah. So I think for at least this first investigation of this data, I would include the lake if it ever had walleye in it. I would only exclude it if now when we look at it, we're like, well, it never had walleye at all. So that's what I would say. So now I'm gonna, and you're saying Crystal in Dakota.
Yeah, I see gill nets for crystal and Dakota for walleye in the year 2000, and that's really about it. Even 2005, I don't see anything. So
But you do see walleye in the gill nets
in 2000, yeah.
in 2000. Yeah, so keep it in the data set. And what we're saying is now the walleye number is 0. Yep, that's what I think. All right. Hey, and Dolly, you have your hand up? You got a question?
Yes. So we have to select one of the 11 lakes, or we're gonna do the same lakes, or what can we do? I'm just
Everybody is doing all 11 lakes.
oh, okay.
And part of the purpose is to get you familiar with using Lake Finder.
Okay, thank you.
That's part of what you're doing is learning how to use Lake Finder, getting comfortable with it, getting so that it's easy to use, and then you start looking at all the other information you can find.
Okay, thank you.
So that's part of it. Yeah, you bet. Thanks for asking that. I'm sure other people had the same question.
Hey, Professor Park.
Yeah.
So I've just kind of been going down the line, down the table, the stocked lakes. For Clear Lake, 3 different lakes come up. Was there one that you wanted us to do?
Yes, let's see which one it is. Clear Lake. One of them is really big. We want the one that is stocked with walleye. I'm sorry. The Clear Lake that we want is the one that is stocked with tiger muskie. So you could go to the Clear Lake and then click over on the stocking report. Washington County. I don't know which one it is. I'm going to try Lake Elmo, the one that's near Lake Elmo. Oh, that's really small and there's no information about it, so that's not it. How about the one that's up by Forest Lake? Yeah, I think this is the one. Let's check. Fish stocking. Yep, this is the one. Oh, I stock it with tiger, muskie, and walleye. That's interesting. That's interesting. So yes, it's this one, the one that is the one near Forest Lake.
Thank you.
And then maybe even, would I even have said it right here? Pass, pumpkin seed. Yeah, here's tiger muskie right here. So, yeah. Yeah, and Dolly, is that a new question or is that your old hand?
It was from previous one. Sorry.
Okay. Do you know how to put your hand down? I mean, I can do it too.
Okay, let me try it. Oh, I got it.
Yep. Bingo, there you go. All right, let's see, get a piece of paper here.
Hey, Virginia, can I just go over the lab with you real quick here?
Sure, the lab?
Yeah.
Okay.
So the stocked table is the lakes where the muskie was introduced. Not stocked is not introduced. We're
Mhm.
gonna look for the CPUE and create the two statistical tests based on the information we get, right?
Yeah, that sounds right. So we're comparing lakes in which the tiger muskie in particular are stocked. And there's only 11 lakes in which tiger muskies have been stocked, so that's the 11. And then we're going to compare the walleye populations in those lakes to the walleye population in 11 other lakes that I just picked to be kind of similar to that first set.
Okay.
And we're comparing the CPUE, the catch per unit effort for walleye. And we've decided that if the lake ever had walleye in it, we're going to keep it in the data set, even if there are no walleye now. And if there is no gillnet data, then we'll have to use whatever it is that they're using now, because they seem to have changed methods. It used to be just gillnet or trapnet, and it seems like now they've got electrofishing and seining. We'll just have to use whatever data is available on the website there.
Thank you.
Okay. Well, how's it going? Seems like,
For
Yeah.
Crystal Lake, they don't have post walleye, and the other one, or what can we do? The tiger.
tiger. So you're talking about Crystal Lake. There's 2 Crystal Lakes in the data set, right?
The Dakota.
And you mean Crystal Lake in Dakota County?
Yeah.
Okay, so Crystal Lake in Dakota County. It does have tiger muskie. And looking at the most recent, it does not look like it has any walleye. I think that somebody said if you go back in time, it used to have walleye, but now it does not.
What about the tiger muskie?
It has
do have?
tiger muskie, but it does not have any walleye. Oh, that's funny. The tiger don't show up in the sampling, but they have been stocked into the lake recently. So, yeah. So we don't need to know how many tiger muskie are in the lake. We only need to know, was it stocked? Were tiger muskie put in the lake or not?
Okay.
So it's kind of, yeah, were they put there or not? It doesn't matter whether they find them again for our purposes.
Okay.
So we're going to say tiger for Crystal Lake in Dakota, tiger muskie, yes. Walleye, 0.
Okay.
All right, how are people doing? Anybody else working? Just working. Okay, so I thought that if it looks like everybody has got this underway, I'm assuming that if you have a problem or a question, you would ask it. So I'm going to assume that everybody is working on the lab and it's going okay. So if anybody has questions about the quiz, I could go over some of those answers. I'm just working out one of the answers right here, right now, so I can write it quickly. Some of the answers, I think it might be most efficient for me to write out an answer and then just post a solution for the ones that involve calculation. But I don't know, if you have a question. So circular lake of area.
Is this for quiz 1 or quiz 2? I think on D2L.
Well, I don't know. It depends on what kind of question you have. I think most people would have a question about quiz 2 because you've already had a week to ask questions about quiz 1. So my guess would be quiz 2 would be the thing people had questions on, but that's just that's my guess, my prediction.
Oh, okay. I think on on D2L, I just saw that you wrote quiz 1, and that's why I just asked.
Oh, I did. Yeah, so you think there might be like a typo on the announcement or something? I can go out there, somewhere out there. Oh yeah, you're right. Okay, I can change that.
Yeah, I had questions. So yeah, quiz 2, question 3.
I didn't, yeah. Okay, quiz, what question is that?
Question 3, it says, which of the following best describes how most lakes in Minnesota were formed?
Oh yeah. I think the key point here, it was a little hard to write that question. It was a little hard to write that question so that it measured what I wanted it to measure. But the point was this: some people think that the water in the lake is left over from the glacier. And that is not true. As you know, the typical residence time for water in Minnesota lakes is months, less than a year. And even worldwide, the average residence time for water in lakes is less than 20 years. So the answer is that the glaciers came down across Minnesota. The glacier ice is full of gravel and sand. And when the glaciers melted away, they left behind a very irregular surface. And in other words, hills and valleys. And those filled with water, excuse me, those filled with water. And that is the source of the lakes that you see on the earth today. And the thing about a kettle creating lakes is it's not really about the ice that's left behind. It's the irregular surface, the rough surface of the earth that is left behind. And let me show you so that was a difficult question for many students. I wasn't surprised by that because in my experience, that's a common misconception about glaciers. I lectured on it and tried to remedy that. Glaciers do not create lakes by scraping the ground, primarily. They don't gouge lakes or dig lakes into the ground. Almost all glacially created lakes, not all, but most glacially created lakes, especially in Minnesota, are actually the result of what the glaciers leave behind. And I'm going to show you this chart, which maybe if you have this image in your mind. Here's the state of Minnesota, and this is the depth to bedrock. How deep beneath the surface do you have to go to find the bedrock? Now, black is 0 to 10 meters. I think this is meters. No, feet, 0 to 10 feet. So down here where the glaciers did not go in the most recent glaciation, the bedrock is not under very much glacial till or soil, neither up here. But for most of the state, all of this green is 100 feet of sand and gravel and clay. And the blue is 600 feet, 300 to 600, up to 1,000 feet thick. So the bedrock, the part of the earth that might have been scraped or gouged, is, tens or hundreds of feet below the surface that you see. And this immense layer of sand and gravel and silt and clay, that immense tens or hundreds of feet, is not smooth and flat. It's actually very, very irregular with mounds and dips and valleys and so on. And that is how glaciers create lakes. Okay, now there are some lakes that are created by lakes scraping things. And there's some lakes that are created by glaciers damming things. And there are some lakes that are created by glaciers plucking stuff out of the bottom, but those are really pretty rare in comparison, especially in Minnesota. Okay, that's my answer to that question. And I hope that this diagram is helpful to you. Here you can see, here, this was not covered by the most recent glaciation. And then here's this other area that diverted the glaciers down there. And the Topeka Shiner lives down here.
I just want to clarify. So the correct answer for that was that the glacier loved hills and valleys made of gravel and sand, right?
Yep. Yep.
Okay.
Virginia,
Can you-
I just had a question for the number 8 and 9 on the quiz too.
Sorry, the what?
Question 8 and 9.
Okay.
Both of those.
Which ones were those?
Those are the equation.
Sorry? Which ones were those? Where did it go?
I don't have it pulled up right now.
Yeah, question 8 is what is the shoreline development factor of a lake?
Oh, the shoreline development factor. Sure.
Yeah.
So the shoreline development factor is the ratio between the actual shoreline and the shoreline that you would have if that lake was a perfect circle. Okay, so in the information that's given for that question, the shoreline development question, right? The area of the lake was 100 hectares. And you got to make sure you convert everything into the same units. So I'm going to convert everything into meters. So 100 hectares, that's 100 times 10 to the 4th meters squared. Because a hectare is 10 to the 4th square meters. And the shore length of that lake was 7 kilometers. So that's 7 times 10 to the 3 meters. Now a circle has area equals pi r squared. So r, the radius of a circle, is equal to the square root of a over pi. So divide both sides by pi, take the square root. Yeah, the radius of a circle is equal to the square root of a area over pi. And a circle has a circumference. A round lake has a shore length, if it's a perfectly circular lake. Has circumference. C equals 2 pi, right? 2 pi r. Because that's a definition of pi, right? The definition of pi is the circumference divided by diameter, right? Circumference divided by 2 times the radius. So that's a definition of pi. Anyway, so circumference is 2 pi r. If you substitute the r in, it's going to have a circumference equals 2 pi times square root of a over pi. I just substituted that in. And you can leave it like that and calculate it using pi twice, or you could simplify it, square pi and put it inside the square root sign. And in that case, you would get so you can use either of these formulas. This one is simpler, so I prefer it. I mean, I prefer to use it because it's simpler. All right, so that's our formula for the circumference of a circle, a round lake that's perfectly circular. So the circumference of this lake, if round, perfectly round, the shore length would be equal to 2 times the square root of area, 100 times 10 to the 4th meters squared, times pi. And then after that, it's just a matter of plugging the numbers correctly into your calculator. I mean, if you wanted to simplify it, you would go you could pull out the 100, the square root of 100 is 10, the square root of 10 to the fourth is 10 to the second power. So this is actually going to be 10 to the 3 times the square root of pi. I'm doing this correctly. And after that, it's just plugging it in to your calculator. I should get a number. Does that explain the answer? The idea is you've got a lake of, area such and such with some kind of shore length, and you're comparing it to a lake of the same area that is a perfect circle. And this might be the measured shore length, and this might be the shore length that's really the shortest possible shore length is the shore length of a circle. And then the shoreline development factor is just the length that you measured divided by the shore length of a circle. So I'm going to take, this is the shore length of a perfect circle, whatever this is. That's going to be down here. And then the shore length that you actually measured, that's going to be here. So the shoreline development factor is kind of saying, well, how long is the shoreline compared to the minimum? If it's twice as long, then the development factor is going to be 2. Now, if it's 50% longer, the development factor is 1.5. It's 10% longer, 1.1. Yeah. Other questions?
Would you be willing to do number 9 as well?
What was number 9?
It was the mean depth, I believe.
Let's see if I can pull that up. Number 2, number 9. What is the average depth of a lake? Oh, the average depth of a lake that is the following measurements. The secret to the average depth is it's, no matter how complicated a lake is, I mean, you might have a lake that's got all kinds of bays and it's got a very irregular basin, it might be some crazy thing. But if you know the area and you know the mean depth, then you've got the volume. So let's say that here's our mean depth. You look at all of that and if you know any 2 of these 3, you can get the third because it's this simple. The volume of the lake is equal to the area times the average depth, the mean depth. So the volume of a lake is equal to the area multiplied by the average depth. It's really that simple. And so if you were given any 2 of these, you can figure out the third. In the problem, you're given the area and the volume. And the question is, what is the mean depth? So the mean depth is equal to the volume divided by the area. The volume was 6 million cubic meters. So 6 times 10 to the 6th million, 1,000 million, yep. 6 million cubic meters. And the area, 100 hectares. And that's 100 times 10 to the 4th meters squared. And it's important, you know, to, if you're going to be doing division, to be using the same units on the top and the bottom. So it's important to convert hectares to square meters. And then you can just cancel. Let's see, 100 times 10 to the 4th, you know, that's actually the same as 1 times 10 to the 6th. And then it's really easy, 10 to the 6th, 10 to the 6th. And the answer is 6 meters. So it really, once you realize how simple it is, then it's really simple. But I guess if you go off on a different tangent, I got some really crazy numbers. I mean, amazingly complicated numbers. I have no idea how people got them. And I'm like, well, I guess that tested if you knew how to get it or not. But I don't know, does that help? Does that answer the question? I mean, does that make sense?
Yeah, it does. Thank you so much.
You know, and if people forgot to, if somebody forgot to convert from hectares to meters, you know, they would get an answer like 0.006 or something. And, you know, I gave half credit for that because I could see like I could see the little mistake that a person made, not converting. But, you know, obviously part of it was done correctly. So I overruled. No, I guess I had to put that in myself. But anyways, I gave half credit for people who did that, who got it at least half right. If I could see what you did wrong, you know.
Mm-hmm.
All right, other questions?
Oh, I just wanted to ask, is there a way we can like review the questions we got correct too?
You know, the way that it's set, the way that the quizzes are set up, you only see the ones that you got wrong, because that's the ones that you need to work on. And I'm not exactly sure why that is considered best practices, but it's considered a best practice with online quizzing. You know, I guess it's kind of like, you know, like with flashcards, you know, you get a stack of flashcards, and when you get them right, you set them aside. And then you just work through the ones that you still need to work on. So that's my answer. I guess when we get closer to the midterm, I may need to reevaluate that. But for now, at least, we're going to stick with, I'm going to follow what I was advised. So for now, we're going to stick with this.
Yeah, when it comes to the when it comes to the midterm, yeah, would you be able to like maybe give the quizzes back or something so we can just study off those as well?
Yeah, I think you'll still be able to see what you can see, right?
I don't know. I can't. I tried to look at it and it wouldn't let me.
Oh, wouldn't let you. So can you see the questions, any of the questions?
Oh, wait. Yes. Sorry. I pressed on the quiz, not the what's it called? The attempt button or whatever.
Yeah, I don't know. And
yeah, I got it though.
And I'm not, yeah, I think that from the student side, it does look a little different than what I see. So, but yeah, so you need to tell me, you know, it's kind of complicated under the hood there. Yeah, Desiree.
Yeah, just one more. Can we go over question 10?
Question 10. Yeah, what was question 10?
It's about table.
Oh, Gorham study? Yeah, that is, best way to do that would be to pull up the table that it's referring to. And I went over the table in lectures, so it must be in one of my PowerPoints. Well, I don't know which PowerPoint it's in. Let's see here. This question refers to table, 7.1 in Calc. So I assume that you have the textbook, so I can just refer to a table and have you look at it. Yeah, here's table 7.1. And when you look at table 7.1, you can see, where's the camera? Here's the camera over here. A bunch of studies reported. One of them is by Gorham. That is about lakes of the world. I think I'm following the right line over here. And then there's an equation. So you look in the table, it says, the question says, look in the table. Find the equation for Gorham's study of lakes of the world. And what you find is this formula. It says regression equation z bar equals 0.46 z max. And then the question is, based on the results of the study, and the results of the study are summarized in this equation, you know, because it says blah, 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 E. Gorham, 1964. And over here it says world. Right, so this line in that table gives you this formula for average depth of a lake. And I think the question was, if a lake has a maximum depth of 100 meters, according to the results of this study, how deep would you expect the lake to be? Well, if our lake is typical of the lakes that Gorham studied, reported here, if it has a maximum depth of 100 meters, 100 meters times 0.46 is going to be the average depth, or the mean depth. So you would expect it to be, if it's 100 meters maximum, you would expect the mean depth to be 46. Does that explain how to answer this question, or how to get the answer?
Yeah, that explains it.
Sorry?
Yeah, that explains it.
Yeah. And so you saw we went over a table today. The table is in the textbook, and the table was in a PowerPoint. So I know that everybody has access to a table. So I could ask the same type of question about the table that we went over today. Based on one of those studies, a lake with a total phosphorus concentration of something or other, what would you expect, you know, the other thing to be? Or I could very easily ask you a question about one of the graphs that were in the, you know, that we went over today. So you've got the graph, it's in the assigned reading. There are reading questions that basically tell you to read the graph. And then in lecture, I went over how to read the graph. So I'm thinking, you know, if the students are learning what I want them to learn, they've learned how to read this graph. So if it's what I want you to learn, it's in the reading question, whatever at lecture, that's a perfect thing for me to put on a quiz to try to see, you know, did the students learn what I wanted them to learn? Not to give away any secrets, but that is in fact how I construct quiz questions, right? I look over the reading questions. I look over my lecture notes. I look over my PowerPoint slides. I think, okay, what did I tell them that they needed to learn? And then that's what I'm going to test them on. So, yeah, it shouldn't be a surprise. And my advice would be, you know, read the chapter, answer the reading questions before class. And that way, if you need help learning something, here we are in class together. This is when I'm available to help you learn it. No, if you only try to learn it before the quiz, it's almost too late, right? You're like, you know, so my advice is totally read the chapter, try to answer the reading questions before class, and then you've got class time in order to learn the things that you realize you couldn't get just from the reading. So that's my advice to everybody. I should probably repeat that when I got the whole class, but you know, anyways, you guys are here, so I know that you're trying to, you know.
Thank you, Virginia. I'm gonna hop off now.
Okay. Well, it's probably time for us to go, but any other questions? I can answer them.